I've already pointed out a couple of times that during the Carboniferous, the imbalance of photosynthesis outpacing respiration and decomposition consistently over tens of millions of years resulted in much atmospheric carbon being converted into biomass and stored away in the form of coal deposits. The word Carboniferous literally means coal bearing, and while coal formation is not exclusive to the Carboniferous, coal is actually produced throughout the fossil record. It's just a fact that the largest conversion of CO2 into biomass into fossil fuels occurred during this time. I've also said that the plants of these massive forests of future coal were the non-seeding vascular plants, like this giant club moss, Lepidodendron, as well as giant horsetails, like this Calamides, as well as giant ferns. Seed plants were already in existence, but they don't appear as dominant forest trees until the Permian. It basically took a full geologic period for this new kind of plant to bubble up through the prevailing forms of plant life to finally make it to the big time. And so while we treat the seed plants as the next great innovators when it comes to plant adaptation to dry land, there's a substantial lag between their origin in the Devonian and their success in the Permian. And so, through the Carboniferous, while non-seeding vascular plants like Lepidodendron were dominating, the seed plants were there, but they weren't just hanging around with a low profile. They were actually diversifying into at least four clades of gymnosperms, plus one other lineage, the seed plants that ultimately give rise to the flowering plants, though this doesn't happen until the Mesozoic. By the time the Permian rolls around, we see representatives from all four kinds of modern gymnosperms: conifers like pine trees, cycads, ginkgos, and a fourth group of gymnosperms, the cnidophytes, which really are nido as far as plants go, though they're not especially familiar to most students. In addition to these known lineages, there were also other kinds of seed plants that showed up and had their heyday as evident from the fossil record, and then they disappeared, leaving behind no descendants. One great example of this is the glossopterids, known from Permian coal deposits in Brazil and southwestern Africa. The distribution of these Permian gymnosperms on either side of the southern Atlantic Ocean was a key piece of evidence used by Alfred Wegener to support his theory of continental drift. He argued correctly that the drifting apart of South America and Africa split into two an area that was once an expansive forest in which Glossopteris was a dominant tree. Now, between familiar kinds of gymnosperms and other now extinct seed plants like Glossopteris, spermatophytes were highly successful, eventually taking over as the dominant forms of large plants on Earth. By comparison with seed plants, the once dominating non-seeding vascular plants were less adept at dealing with dry conditions. And in fact, the modern representatives of those Carboniferous giants, modern horsetails, ferns, and club mosses, these are plants we associate either with wetlands habitats or moist and shady conditions. And so this brings up the question of what is the advantage that seed plants have over their non-seeding antecedents in persisting better under drier conditions? Start by picturing the alternation of generation cycle of a fern. An adult fern sporophyte, it can grow successfully under relatively dry conditions by accessing soil moisture with its roots. It doesn't require a wet soil surface. But in order to reproduce, its spores must land on a wet surface, grow into prothalli, that is gametophytes, and then sperm released by some gametophytes have to travel to other gametophytes to fertilize eggs in their archegonia. And then the next sporophyte has to grow and establish enough of a root system to access the deeper soil water. And all this has to happen before the surface of the soil dries up. So while a fern sporophyte can enjoy the benefits of being vascular, sending down roots to capture deeper soil moisture, the gametophytic stages of these plants seem more similar to algae, lacking entirely in vascular tissue and therefore requiring wet soil, if not complete submersion under the water. It's not surprising that a fern's natural habitat is typically one where you have seasonal standing water or at least very wet soil for a long enough period for all of those intervening stages to complete. Basically, a fern is like a frog in not being fully independent from its aquatic past. It's forced to return to an alga-like state as a gametophyte, just like the frog must return to a fish-like existence as a tadpole larva. Now, all of the non-seeding plants, ferns, horsetails, club mosses, quillworts, they all do things this way, and it's presumed that those enormously successful non-seeding plants in the Paleozoic also required semi-aquatic phases in their life cycles. The seed plant's great innovation here was to eliminate the need for that extensive wet soil period. They do this by creating moist microhabitats within their sporophyte bodies in which the spores can develop and mature into adult gametophytes, all while still attached to and receiving nutrition from the parental sporophyte. Now, an important part to understand here is that the strategy employed by seed plants requires there to be two kinds of spores, giving rise to two kinds of gametophytes, one male and one female. The adult male gametophytes are the pollen grains. It's their job to travel through the air and then deliver the sperm to the female gametophytes. These male gametophytes, again, get it through your head that each pollen grain is a fully mature whole multicellular plant, the gametophytic plant that grows from a unicellular microspore. These haploid microspores are produced via meiosis and matured within the microsporangia, which, if you remember from your Latin lessons, means vessels containing microspores. Microsporangia are located on specialized reproductive structures known generically as microsporophylls, literally leaves of microspores. Now, there are specialized names for the microsporophylls depending on the kind of plant you're looking at, but we'll get to this later. For now, here's a rundown of the basic terminology, and I hope you see the pattern. Microsporophylls are the specialized leaves bearing the microsporangia. Microsporangia are the vessels within which the plant undergoes the meiotic cell divisions that produce the microspores. Microspores are the unicells that undergo mitotic cell divisions and development, ultimately culminating with the maturation of the adult microgametophyte, which is the pollen grain. Yeah. The same array of specialized structures and cells is there on the female side as well. Megasporophylls are specialized leaf-derived structures that bear the megasporangia, vessels containing megaspores. Each megaspore, which is a unicell, undergoes mitotic cell divisions, developing into a mature multicellular megagametophyte. There are two things I want you to visualize here. First, the microgametophyte and megagametophyte are analogous structures to the fern prothallyi that we saw under the microscope in laboratory. In the wild, these fern microgametophytes live independently as little green flecks in the moist soil. Under the microscope, you will see a scrawny male gametophyte with only antheridia. Now, visualize this scrawny male haploid consisting of a few hundred cells, becoming further and further and even further reduced to the point where all you have is a couple of cells, of which one is the sperm. This is your pollen grain. In the fern, that scrawny male released its sperm cells, which then swam to locate a female with an unfertilized egg in its archegonium. Now, the pollen grain does things differently. The whole microgametophyte floats through the air to reach the female structure. And there the sperm is delivered to the egg, but the distance covered from the release point to the fertilization site is traveled through the air by the whole gametophyte, and not swum through water by the haploid gamete. Meanwhile, on the female side, instead of being independent like a little green fleck in the dirt, as is the case with a fern, the seed plant's megagametophyte remains within the moist refuge provided by the sporophytic parent, surrounded by nurturing tissue on the megasporophyll. The pollen arrives basically at the megagametophyte's doorstep and delivers the sperm cell more or less directly to its egg. Fertilization ensues, and the sporophytic parent continues to nurture the gametophyte while it and its zygote grow and develop into what we call the seed. The zygote formed from the fertilization develops into the embryo of the next sporophytic generation, while the haploid tissue of the gametophyte becomes a storehouse of carbohydrates and fats. Typically, this is the bulk of the seed. Ultimately, the seed will disperse from its parent plant, and with some luck, it'll germinate. And at that point, the nutrients stored will be feeding the baby plant. Now, I can say from experience that most students require a few passes through these ideas in order for it all to click and make sense. Not because the material is especially difficult or complex. It really isn't. The difficulty that students have comes from this all being so different from the way that animals do things. Now, it's also going to help tremendously if you've already mastered the concepts of alternation of generations, because this is all about keeping track of that gametophytic generation. Now, the second key idea here is heterospory. You can say heterospory, that's okay. You'll note that the scenario of seed plants requires that there be two distinct kinds of spores, megaspores that become megagametophytes and microspores that become microgametophytes. This represents a central departure from earlier plant forms. Ferns, horsetails, lycophytes, mosses, and liverworts, they're all homosporous, producing only one kind of spore. Heterospory, literally different spores, is a necessary precursor to the evolution of seed plants. You can look at heterospory as an extension of something that we saw in the laboratory, where the ferns produced some protheli that were exclusively male, bearing only antheridia and carrying only sperm, while other protheli were female, bearing only archegonia and carrying eggs. But the ferns in which we saw this were homosporous. All the spores they produced were identical, with the same potential to become either male or female. The sex of the gametophytes growing from the spores was determined by the growing conditions. Under good conditions, the prothallus grows up nice and strong and develops into a female. Genes for making antheridia are turned off, and the genes for making archegonia are turned on. Now, under less than ideal conditions, the prothallus grows weakly into a small, pathetic little thing, and that's the signal for the antheridia genes to turn on and the archegonia genes to turn off. Scrawny gametophytes are the male gametophytes. The separation of male from female in fern protheli had a couple of logical significances. For one, it's fully expected that not all spores are going to be able to grow up into full-size gametophytes, and yet it's only the large gametophytes that have any chance of being successful at nurturing a baby sporophyte. Remember that the resource that will give to its baby is whatever is already there in its body. For a small, dinky, scrawny prothallus, there's really no pathway to success in producing archegonia and eggs, and so it makes perfect sense for them to allocate all of their energy towards sperm production. The other side of this argument was that being dioecious as gametophytes means that there's no possibility for self-fertilization to occur within a single prothallus. No chance for sperm fertilizing an egg from the same gametophyte. If such intragametophytic self-fertilization were to happen, this would guarantee exposure of lethal or deleterious recessive alleles, and the resulting sporophyte would have basically zero fitness. By having gametophytes be either male or female, this becomes a non-issue. Now, homosporous ferns achieve separate sex gametophytes in the way that I just described. Heterospory achieves gametophytic dioecy by predetermination at the point of spore production. In differentiating cells to become a microsporangium, the plant is specifying that the microspores produced there will develop only into pollen grains. Meanwhile, on the megasporangia, a different developmental script is followed, resulting in much larger megaspores that develop only into much larger megagametophytes. The differences are substantial. Microgametophytes, the pollen grains, are released by the microsporophylls, and often in enormous numbers. Anyone with allergies knows all about this. Megagametophytes, on the other hand, are not released, at least not right away. They remain on the megasporophyll through fertilization and beyond, nurtured by the layers of integument that form a placenta-like connection between the megagametophyte and the sporophytic parent. Ultimately, the seed develops from a complex of haploid and diploid cells that were there. You have cell lineages descending from the haploid megagametophyte, separate from the diploid embryo that develops from the zygote. There's also quite a bit of diploid tissue deriving from the sporophytic parent. In effect, there are three generations represented in a seed: the sporophytic grandma, gametophytic mother, and sporophytic granddaughter, all there in different parts of the seed. Pollination is achieved with the arrival of a pollen grain, that is, the mature microgametophyte, to the megagametophyte. Delivery of the sperm to the egg is the next step, and this can occur in one of two ways. In the gymnosperms, cycads, and ginkgos, we have this method where the pollen grain gets drawn into a liquid-filled space, the pollination chamber, which contains kind of a sugary water solution. There, a motile sperm cell erupts from the microgametophyte, and then it swims a very short distance to reach the egg. In conifers and neophytes, as well as the flowering plants, the pollen grain does this other thing where it sprouts a pollen tube that grows right up to the megagametophyte, penetrates the micropyle doorway, and delivers a sperm nucleus directly to the egg. With a fusion of egg and sperm, a diploid zygote is formed, and this is the part that develops into the embryo of the seed, which eventually grows into the next sporophytic plant. The rest of the megagametophyte remains haploid and develops into the part of the seed that will provide energy to the young sporophyte after the seed germinates. In this sense, the original embryophyte condition is preserved, gametophytic mother nurturing the early growth of her sporophytic offspring by providing energy from her own body until the daughter is able to live independently. Seed plants are embryophytes just as much as mosses, and they use alternation of generations just as much as a fern does. All the parts are there, but the bits are much more derived and sometimes kind of hard to recognize. You should be able to trace the gametophytic and sporophytic generations here just as you did for mosses and ferns. Make special note of the important differences from the non-seed plants. There's no semi-aquatic stage, and these plants are heterosporous. Now, what were some advantages that seed plants had over the non-seeding vascular plants like ferns and lycophytes as things got drier in the late Carboniferous and Permian? The kernel of this lesson is the issue of how seed plants continue the trend of plants increasing adaptation to dryland habitats through the Paleozoic. In fact, you can see that over the Paleozoic, plants make three important innovations in sequential order that culminates with the success of seed plants in the Permian and beyond. In the Ordovician, plants make their first move on a dry land thanks in part to the evolution of sporopollenin and the embryophyte condition. These innovations allowed plants to invade into what had previously been nearly completely barren terrain, the dry land of continents. In the Silurian, we see the earliest fossils of true vascular plants, the Eutracheophyta. Vascular tissue, the reinforced water transport tubes, allowed plants to mine for water and minerals below the soil surface and to grow into bigger plants. The success of this innovation is evident from the Carboniferous forests. Global domination by plants operating on the basis of photosynthesis and vascular tissue only. Heterospory and seed plants, with their final declaration of independence from their aquatic past, was the third and final innovation that we see during the Paleozoic. The ability to complete the entire cycle of alternation of generations without involving a free-living semi-aquatic gametophyte allowed plants to move into areas that weren't seasonally flooded. In the next video, we return to the phylogenetic side of things, adding further taxa as we move out into the more derived branches of spermatophytes, plants with seeds.